Welcome to MOOC NPTEL course on Bioengineering, an interface with biology and engineers. In today's class, we will study genetic recombination and linkage, the production of offsprings with combination of traits that differ from those found in either parental generations. Morgan, in addition to making contribution for understanding the chromosomal basis of this inheritance, he was also making some many interesting fundamental observations. If you now think about going back to meiosis, at some point we have said that the genes get recombined, and that's what gives lot of uniqueness among the progenies, right? Among the child you see lot of differences because the genes get recombined. So he was observing this, that whether the genes recombination event happen, or there are some genes which are very closely linked to each other and they are not getting recombined. So the production of offsprings with combination of traits which differs from the parents, whether those are passing together or they are getting segregated. So think about the classical Mendel thing first, and then we come to Morgan. So in Mendelian experiment we have seen these are the parental type offsprings, and then when we did this dihybrid cross, we also saw some recombinant offsprings appeared. Recombinant means some new type of features appeared along with the original combination of the characters. So two non-parental phenotypes were also seen in the offsprings, and these are known as recombinant types or recombinants. So if you remember from the meiosis image, these chromosomes are exchanging a small segment of it at a site which is known as crossing over. So from this part, from the male and female gametes, there are certain interchanges happening, and now you can see in the recombinant A and recombinant small a, this part is actually transferred. So all the genes which are part of this one has now come in this chromosome, and all the genes from this part has come to this chromosome. So this is kind of concept which we have briefly seen in the meiosis part, and Morgan was trying to investigate this in much more detail. So just to refresh you, recombination is an event when these genes are getting recombined and they are getting exchanged from these two chromosomes, and at a site which is known as the crossing over site. All right, so yeah, this part we have talked in the meiosis prophase 1, just to be precise, and we have seen that a set of proteins are getting exchanged as a part of it, which we say as a crossing over event. So how does, if the genes are linked or they are far apart, how does that information can be transmitted from the one to next generation? And Morgan was always using the fruit fly as a model system, and he wanted to study these investigation also in the fruit fly. So let's now increase a little bit more information from the fruit fly experiment. And in this case, now we have two different type of fruit fly. One is wild type, which is having the gray body and the normal wings, and second is a double mutant, which is black body and the vestigial wings. Just to simplify this part, if you make these kind of crosses or two properties like this, that if we are saying wild type, it means it is plus, right? And we are showing on both the chromosomes, so B plus and VG plus, B plus VG plus, and we are doing a cross with double mutant. So what is shown here, B plus is it means showing on both the chromosomes, right? So this is how the cross should ideally look like. And so now if you take this situation that you have a wild type with two features, and what you are looking at is studying two genes, and are they linked to each other, or are they going to separate in the process of recombination? Now in the F1 dihybrid cross, all of them are looking like the same, the wild type feature what we had. And now if you want to do a test cross from this, think about Mendelian test cross. So what would you do the cross now? If you want to do test cross, F1, whatever is the phenotype shown here, this F1 you want to do a test cross. So what will be the genotype of the other fruit fly? You want to do a cross of this particular fruit fly and you want to do test cross. So what we did for a test cross in Mendel's PE experiment? Anybody recall? Homozygous recessive. Is this homozygous recessive? This is right. All right. Well, so now can we see this thing? This wild type of F1, and we do a test cross with homozygous recessive double mutant. Please do that. Write down these gametes and make this square. So now you can write these gametes. So how this will look? Okay. Now you try other.
You are trying to do the test cross. So what does it mean? Like you are saying that there are 4 different types of eggs which will produce, which you will cross with the test cross. But
let's discuss after doing the cross first. All right, and now if you can read from here, these are gray body and normal wing, and this is black body and vestigial wings. Could you also write down what will be the phenotype of these flies? Write down what are the phenotype of these flies. Plus means wild type, and
when we write B plus,
yes.
then we consider that B plus is dominant.
So that's why I was telling that in all of these problems, it's a good idea to write B plus, VG plus, and B and VG, because these are like this, because these are on different chromosomes. So it's ideally the same thing. But when it is plus here, it is mean it is wild type, and if it is not plus, it is mutant. So we have now these 4 different type of phenotypes, and what we are observing here that one is gray normal, one is black normal, one is black vestigial, and one is gray vestigial. So in addition to the 2 which were original phenotypes what we had, we have 2 different new phenotypes as well. So what Morgan was trying to understand from the experiment, whether these genes are very closely linked to each other or they are very much far apart. So if you think about, just imagine that on this chromosome, if this part is getting recombined, and these 2 genes which we are studying, one is this part and one is going to get separated, it means these are not linked. But if these 2 genes are here or these 2 genes are here, these are going to be linked, right? If they are very close to each other. So then if they are close to each other, so even if they are getting recombined, still they are transmitting together. So therefore these 2 genes, a higher proportion of this looks like they are linked to each other. But there are some progenies, some flies which were also found of other phenotype, which may be that some recombination event might have happened. So he was the first time he was looking at not only that how genes are being transmitted, but where they are located on the chromosomes and are they linked to each other or not. So this is where he was looking at the linkage phenomenon, and what he concluded that most offsprings had parental phenotype, whereas very small number, relatively very small number of the flies were also having the non-parental phenotype, which means that there might be some mechanism which breaks this particular type of linkage, and some recombination events might happen which is being transmitted from one to next generation, which happens usually during the meiosis process. All right, let's take a short break and let me show you a clip. It's not going to be a quiz question, but what is this movie? Very good. And what is the character? What is this character? Character name? Oro. And what is this disease come from? From which syndrome? That will be a question. All right, so I'm sure you would have appreciated when you would have at least watched the trailer of this movie that there are some child who might be suffering from these kind of chromosomal glitch to say, and those sometimes could just be because of one gene defect. And in this case, it was based on a lamin A gene, which is defective. So there are many diseases which happen because of certain alterations happening at the chromosome level. And let's kind of not discuss some of these kind of genetic disorders which are happening because of abnormality in the chromosome number or the structure. So when we want to study about all the chromosomes, if you think about at some part of developmental study, we talked that you want to map the karyotypes, you want to look at all the chromosome patterns, and it's just color coded differently for each pair of chromosomes. So it is possible that while various recombination events are happening, various genetic events are happening, some part of chromosomes get structurally changed. And it don't happen always, and don't happen very often, but once in a while it happens. And those may happen that in a small part of the chromosome get deleted. And in this case, this D part is deleted. So this is known as deletion of the chromosome. So all the genes which are a part of that part of the chromosome is going to be absent. Or it can be inverted. For example, if the gene sequence was A, B, C, D, E, now B, C, D became inverted. So now you have A, D, C, B. So many of the cascade of the gene signaling will get changed because of this inversion part. Or a segment of it, like BC in this case here, gets duplicated. So now you have 2 copies of B and 2 copies of C. One more situation is translocation. A part of these chromosomes move from one to other chromosome, like AB in this case moved to MNO, and MNO moved to AB. So now they became very different characteristics. Or sometimes it can be non-reciprocal. It means it is just going to get attached to other chromosome, not the vice versa. So there are many things which can happen at the chromosomal structural level, and these are all different type of aberrations which result into different type of diseases, different disorders. For example, you see this child here suffering from Down syndrome. One of the very commonly found syndrome, which is having 3 copies of this 21st chromosome, which is known as trisomy. Trisomy means 3 copies of this chromosome. So Down syndrome is usually result of an extra 21st chromosome. So now their total chromosome number will be 47. So there is a disbalance, and these trisomy of 21 is now people do test for this at the prenatal level for testing whether the fetus might be carrying this syndrome or not. So another syndrome which is something very similar, which is trisomy of 18 chromosome, that is known as Edwards syndrome. You can see this particular phenotype here, Turner syndrome, which is based on absence of the certain genes from the X chromosomes. And now here you can see a phenotype for Patau syndrome, which is a trisomy of chromosome 13. So 3 copies of chromosome 13 is present in this case. So many of these things are some abnormalities which happen because of the way chromosome would have shown some changes at the structural level. And we have seen this example that even sometimes a specific gene like lamin A gene in this case having some mutation, and as a result it resulted into this Hutchinson-Gilford progeria syndrome, and then this kind of child is being born, which now people can do genetic testing and try to find out these kind of genetic abnormalities beforehand. And that's where our understanding of molecular biology and lot of genetic tools are becoming really important in medical field. But what happens at the plant level? So many times plants also show these kind of changes, but not always these changes are very detrimental for plants. So, in plants what is more common occurrence is that certain chromosomes just totally get duplicated, or they make 4 copies or 6 copies or 8 copies, and this phenomena is known as polyploidy. You have multiple ploidy level of those chromosomes. So, rather than only one chromosome making extra third copy or fourth copy, their entire chromosome set has become duplicated. And this is what is shown you here, that 2n, 4n or the 6n of the whole chromosome sets. Look at this particular watermelon. I am sure you would love to eat watermelon without seeds, right? Seedless watermelon. And those are actually polyploids. Those are having extra set of chromosomes, and sometimes those result into some beneficial properties in plants. For example, many of the high yielding wheat varieties, those are hexaploid wheats. So, it's not always that if you have some extra chromosomes, those are going to have problems. In this case, it is much more advantageous. But what is important here to note, in plants we have full set of chromosomes getting duplicated, and that's usually is much more easy to retain for the cell as compared to only one chromosome getting extra. So, that's where I think you can see these are still giving us some positive results in the plants. Let us now solve few sums for genetics problems based on this concept. But before that, these questions will help you revise your concepts. If you do not get this right, please refer back to the slides before you begin the problem sums. Determine the sequence of genes along a chromosome based on the following recombination frequencies: AB 8 map units, AC 28 map units, AD 25 map units, BC 20 map units, BD 33 map units. The further apart two genes are, what will be the probability that a crossover will occur between them, and therefore, what will be the recombination frequency? Chromosomal abnormalities occur due to missing, extra or irregular portion of chromosomal DNA. Which of the following would not disrupt genetic balance? Let us now begin with sums based on chromosomal basis of inheritance. My TA will assist you in this process. In some dogs, the gene for calico multicolor is codominant. Females that receive a capital B and a capital W gene have black and white spots on brown coats. Males can have black or white spots, but never calico. Here is what a calico female's genotype would look like: capital X, capital B, capital X, capital W. Show the cross of a female black dog with a male white dog. What percentage of the pups will be calico and female? Red-green color blindness is caused by a sex-linked recessive allele. A color-blind man marries a woman with normal vision whose father was color-blind. How many F2 plants of each of the 4 phenotypes do you expect? genetics experiments. Thank you and see you next week.